Good afternoon. My name is Paulina, and I'm a second-year PhD student at the Laboratory of Modeling of Cellular Processes at the University of Warsaw in Poland. I'd like to tell you about GPCRVS, a web service developed recently at my lab. The GPCRVS is an online decision system that predicts compound activity against a number of G protein-coupled receptors by using machine learning algorithms and structure-based virtual screening. The first machine learning algorithm in GPCRVS is an artificial neural network. These are computational models inspired by the structure and function of the human brain. They can be used for image or pattern recognition, clustering, or binary and multi-class classification. Neural networks consist of interconnected nodes, or neurons, organized into layers and connected to each other via edges, which model synapses. They have an input layer, one or more hidden layers, and an output layer. Signals pass from the input layer through the hidden layers and to the output layer. The second machine learning algorithm in GPCRVS is based on gradient boosting decision trees implemented using LightGBM. Gradient boosting is a machine learning technique that is used for both regression and classification tasks. A model is built by sequentially combining multiple weak learners, usually decision trees. The idea is to iteratively improve the model by focusing on the errors of the previous models, which is achieved by training each new model to predict the residuals of the combined ensemble of the previous models. G protein-coupled receptors, or GPCRs, constitute the largest group of cell surface receptors and regulate the majority of physiological processes. They transduce signals via either G proteins or beta-arrestins. GPCRs are of particular interest in pharmacology since they are the target of over 30% of currently available drugs. Certain GPCRs, classified as immune receptors, regulate innate immunity and thus represent important drug targets in new antiviral, antimicrobial, and immunosuppressive pharmacotherapies. Unfortunately, many GPCRs still have unsolved structures which can hinder in silico drug discovery. In such cases, ligand-based methods can be used, which do not require the structure of the target protein, but rather a database of compounds known to bind to it. In our web service, ligand-based virtual screening is implemented through a TensorFlow deep neural network and LightGBM gradient boosting decision trees. We first tested our machine learning pipeline on chemokine receptors. Chemokines are small proteins that participate in the transport of leukocytes to injured areas of the body. They're promising drug targets for the treatment of infection, allergies, autoimmune diseases, vascular diseases, and allograft rejection. Through a combination of our two machine learning methods with structure-based virtual screening and molecular dynamics, we were able to propose novel active compounds for receptors CCR2, CCR3, and CXCR3. The machine learning algorithms were trained using datasets consisting of the active compounds of several GPCRs, including chemokine receptors. For these receptors, we retrieved the IC50 datasets from Campbell, which included the SMILES and IC50 activity values of the compounds. LightGBM predictions were found to be more accurate when inactive compounds were removed from the datasets, as you can see in the plot on the upper right. The predicted activity values were highly accurate, as shown by a trend line in red. In the column chart below it, you can see that the number of correctly predicted compounds is also slightly higher in comparison to the model trained with inactive compounds. This means that for multi-class classification or regression tasks, the removal of negative data points improves the prediction. Negative data points, or inactive compounds, are needed for binary classification to distinguish between inactive and active compounds. LightGBM provides continuous activity values for activity predictions, in contrast to the discrete ranges of activity predictions provided by the TensorFlow neural network. In tests performed for the chemokine receptor CXCR1, you can see that the activities of known drug candidates were predicted more accurately by the neural network, while the opposite is true for CXCR2. In addition, especially in the case of the neural network, compounds with p-Campbell values above around 5 are predicted more accurately. This is the main page of our web service. It contains the form for uploading the compound you want to test. Compounds can be either a small molecule or a short peptide. You can choose whether you want to obtain the orthosteric or allosteric binding mode of your compound, or both. Though most of the receptors included in GPCRVS are Class B receptors for peptide hormones, chemokine receptors CCR1 through CCR6 and CXCR3 were recently added. Most receptors are included in both an active and inactive conformation depending on the availability of their structures in the protein data bank. An overview of the predicted compound activities is presented in the form of compound profile. This compound profile shows for which GPCR the compound activity is the highest and to which GPCR it binds with the highest affinity. Lines in the plot correspond to predictions in TensorFlow, gray, and LightGBM, black. The colors of the columns represent the AutoDog-Vena results for the binding affinity. The predicted activity of the tested compound is shown as a red line and is juxtaposed with experimental activities of known compounds of a certain receptor. With a highly active compound, the red line crosses through the points indicating the results for the known active compounds. Before I end my presentation, I'd like to thank all the people who took part in the study and contributed to the development of GPCRVS. I'd also like to thank the National Science Center in Poland for funding this research and PL Grid for providing computational resources. Thank you.